At this time, I'd like to turn today's program over to Ingrid Miller. Ingrid, the floor is yours.
Thank you, Emily. I'd like to thank everyone for joining us today, and I'd like to introduce you to our presenter, Julianne Zedalis. Julianne has taught AP Biology, Forensic Science, and Anatomy and Physiology for more than 25 years at Albuquerque Academy and the Bishop School in La Jolla, California, where she holds the Bobrovsky-Hinshaw Endowed Chair in Biology. She has received numerous teaching awards, including the Siemens Award for Advanced Placement Teaching and the Presidential Award for Excellence in Science and Mathematics Teaching. After her AP Biology course at Bishop's was ranked first in the College Board's Report to the Nation for 2 consecutive years, Julianne was selected to serve on the AP Biology Redesign Commission. She was lead writer for the course's new 2012 lab manual, AP Biology Investigative Labs and Inquiry-Based Approach, and has explained the changes to the AP Biology curriculum at several professional conferences, including APAC, NABT, and NSTA. She also authored a curriculum module, From Gene to Protein, a Historical Perspective, that is available to teachers attending professional development workshops, and a course planning and pacing guide for the revised course that is available online at AP Central. Welcome, Julianne.
Hello, everybody. Hello, everybody. This is Julie in San Diego, and first of all, I want to welcome you all at the end of probably what's a very hectic day at the end of a school year, and we really appreciate that you can join the presentation. We thank Bio-Rad for setting this up for us. When you consider that about 8,000 to 9,000 teachers teach almost 200,000 students in AP Biology, it really is a great way for us to be on the same page with the changes to the new AP Biology course. So this afternoon, whether you are a first-time teacher, you're going to be teaching AP Bio for the first time this fall and definitely have a little anxiety how in the world you're going to get through a 1,000-page textbook, or if you're the more, I call them the more experienced teacher, and you think, wow, you know, I really don't want to throw out the curriculum that I've been using for the last 10 or 15 years, this webinar is for everybody, and we're going to show you how to set up a course, how to design your instruction. We're going to talk about the changes to the lab program and the philosophy behind those changes, take you through the new curriculum, show you the transparency, and try to relieve a lot of anxiety that we're all on this journey together. And I know for me, starting with this project in 2006, it really has been that, a journey. And I can honestly say that people who know me, I've evolved as a teacher, pun intended, through this project, and I think it's going to take all of us. It's a grassroots movement. And we're going to just, you know, be a student of all of this and have fun with your kids, and we'll get through all this. So we've got 4 learning goals. Show you the overview of changes to the curriculum, how you can adapt your current curriculum or syllabus, or how to set up a new one to the new curriculum framework. Show you some tips and websites for professional development, and then at the very end, a little treat. I'm going to address changes to the new exam. And if you still have any brain left, you can even try to answer some of the new questions kind of thing. And to start off, I want to apologize. We are all teachers, and I'm violating one of my #1 rules in my own classroom, that I should not be talking more than 20 minutes at a stretch, because after 20 minutes, students tune out and turn off. But unfortunately, you guys are going to have to listen to me for about an hour, and then we'll save some time for questions and answers. So I'll try to make this as engaging and interactive. You know, I wish I could make it more interactive, but we have what we have. So what we're changing and why. This is not just something the College Board decided, oh, let's change the curriculum. There are many AP courses that are being looked at. And, you know, if you look at your textbook, the introductory textbook, you know, for most of them for AP Biology, I know mine is at least 1,000 pages, and it seems to grow every year as obviously we, you know, have new information that's coming in. And so really over the years, the course had become about 1 mile wide and 1 inch deep. So in 2002, the National Research Council recommended that the primary goal of AP should be to help students develop a deep understanding of the organizing concepts and principles in biology. These are the big overarching concepts that any kind of new curriculum has to be centered about. Also, the curriculum for advanced study should emphasize, and this is a key, depth of understanding over exhaustive coverage of content. So definitely we have cut some content to get that deeper level of understanding, and we'll show you how you can do that. But that doesn't mean that, if you don't see some of your favorite things or your passions on the new curriculum framework, that you have to toss all that out. Show you how you can incorporate your favorite things into the new course. And then finally, what is a reasonable number of concepts that can be studied in depth by students? And, we see in our course that, that's interesting, but I'm sorry we have to move on or we're never going to finish the course in time for the exam. And especially at the end of the year, it seems like we're all racing this amazing sprint to the finish line. This changes to the AP Biology course. We really are, front and center stage. All eyes are on us, and they have been on AP Bio as far as leading the sciences for a very long time. And we have received a lot of really positive publicity about the changes. For example, in 2009 in September, Bruce Alberts, the editor-in-chief in Science magazine, addressed that the changes to the AP Biology course represent major reform in science education. In fact, at a conference, one gentleman said, if we really look at it, these changes to AP Bio are probably the most revolutionary changes in science education in the last 50 years. Also, we were featured in the New York Times, the education section in January 2011, that the College Board took the criticisms to heart, and it seems like we've got it right, or we're well on our way to getting it right. Validation study. This project involved hundreds of people, hundreds of thousands of hours, lots of dollars. When we thought we had a curriculum in place, it was validated by at least 60 department chairs and faculty at leading universities and college that have really complemented the new and improved curriculum, has met its stated goals. That the course will indeed prepare students for success in sequent college biology courses. The breadth and depth of the new course are appropriate for 2 semesters of college biology study. And over the last decade, we have found, fewer and fewer colleges giving credit for scores of 3 and maybe even some colleges with 4s. But now that everyone is on the same page, it appears that the new course is highly favorable for granting credit placement. And as we're going to see tonight with the transparency of the curriculum, no longer are teachers going to have to guess, when it says your course should be covering, should be 20% molecules and cells. What exactly does that mean? It just says evolution 20%. What about evolution? How does this tie into the test? How does this tie into the lab? Now everything is transparent. And at one conference about 2 years ago, a department chair from a Midwestern university and stood up and she said, now that we're all on the same page, she said, when we know a student comes to our school and they received a 5 on that AP exam or a 4 or a 3, it doesn't matter if they're from an urban school, a suburban school, a private school, a public school, a charter school, that student has learned an awful lot of biology, and we know what biology that student has learned. So I think this validation study was very, very important. How do you reduce breadth and increase depth? And this was the first challenge that was given to us by the NRC and the College Board. We would not have a redesign if we were just somehow reorganizing all the content that's been accumulating in the course over the past, probably more than a decade. So for example, just my own course, I am sort of wedded to the Campbell textbook, even though I, pick and choose from a lot of different textbooks. But for example, out of the Campbell 7th edition, for my own course, I found that content in about 20 out of 55 chapters has been eliminated or reduced considerably. In the curriculum framework that we're going to be looking at in a few minutes, we're going to see content that is required, content that is illustrative, that yes, you can include it to, add flavor to your class or add that depth. But if anything is illustrative, it cannot be tested because it means that, teacher A may be including this and teacher B might not. Where I know I'm going to save a lot of time to be able to, get more involved in the lab projects or investigations is down here in these chapters, we jokingly call about a third of the textbook March Through the Phyla, where literally every day we're taking another organism and, going from the jellyfish to the earthworms to the vertebrates, so on and so forth. And also down here at the end that we all, many of us that follow sort of a traditional path through the course, it seems like at the end of the course we're reduced to organ of the day, where, yes, today we're going to talk about the circulatory system, tomorrow we're doing the kidney, the day after we're doing the nervous system, and I know you have lots of questions and we could investigate a lot more things, but I'm sorry we only have one day and we have to move on. So in the new course, the only required systems of vertebrate animal form and function would be the immune, the nervous, and the endocrine, and even that content, what details about these systems is going to be described in this document. I will show you the curriculum framework. Now, this does not mean that you can't teach the circulatory system. I will never give up the circulatory system. But if I can tie it to something else, and if you really, have great activities that you really want to include, the kidney and the relationships and homeostasis, absolutely you can do that. So the first document, to give you a little bit of homework for this summer so we can look at this as summer reading. If you do not have a copy of this, or if you are unfamiliar with the curriculum framework, there is a document that you can download, print out a copy right now. I think eventually it will be available in hard print to purchase. But the most helpful website, or one of them, is AP Central at the College Board. And if you get onto that website, you don't need a password. You just hunt for, it's pretty easy to find, Visit Advances in APs. And on that site, you can access this amazing course and description booklet that is called this Course and Exam Description. And also within here, you'll be able to get the new lab manual, the course planning guides, the sample exams. So the first thing to start off, I think, with your homework is visiting this site. And I think it's definitely worth the paper that you would use to print out a copy of the curriculum framework. I carry like 3 of them around, and every time I'm thinking of a new activity or a new way to do something or a new question, I can refer to that. After about 6 years, I practically have this thing memorized, unfortunately to say. As my own children says, Mom, how many full-time jobs can you have here? But this really is where we started. So we have this wonderful document that we call the curriculum framework, and this is where the absolute transparency is now here. And this has took about a year of a dozen people working with the College Board, several trips to New York and Atlanta to work this thing out. I even brought the committee out here to La Jolla for one meeting just to move us, not all the East Coast, let them do a little flying west. And so we really had a look at, if we just know we have to cut some content, what are those main concepts that every student taking an introductory college course in biology should be exposed to, and what knowledge should they leave that course with? So we were, just one day we talked about, what are those big ideas? And as cheesy as maybe it sounds, we did base the curriculum on what we call the 4 big ideas that we'll flesh out in a few minutes. Then under those big ideas, what are those enduring understandings? What do we want kids? What do we think is important? We know that structure and function relationships, homeostasis is important, evolution, use of energy. Under that, to go to a little finer grain size, what are the essential content or knowledge pieces that you will have in your course to support the enduring understandings and then the big ideas? Something new that has not been spelled out. We all do science, and you're going to already be doing some of these things with your students that are called the science practices. These are skills. What should a student be able to do? How does science work? And we're going to look at those. If you start merging content with these practices, we're going to have what are called the learning objectives, and I'm going to show you this evening how these are constructed. These are very important because it's these learning objectives that provide the framework or the foundation for the test developers and test writers. So you have access to these learning objectives in the curriculum framework, and the Educational Testing Service is working with the AP Biology Development Committee. We are working together when we write these test items that we also are basing items on these learning objectives. So for example, if you just take the test that the kids just took on May 14th, kids said question number 3 was really, really hard because there was a lot of information in there about gene regulation. And for example, siRNA, you may not have known whether or not that should be discussed in class. Where's that depth? How did you know that maybe you should be covering that? So now in the curriculum framework, all that is transparent. The year before, the concept of water potential and for students to be able to manipulate that equation. All that information now that is required is spelled out in these learning objectives. So there should be no guesswork anymore for any of us. Geez, I wonder what the questions are going to be this year and what do you think they're going to focus on? Then also within this document, again, here are some of these illustrative examples. These should be very important to you if you're just starting off teaching this course and thinking, wow, you know, there's so much I could talk to, you know, to the students and so much information I could give out. Where in the world do I even start? So these will give some samples, you know, and examples of where to go with that. And for the first time, we have also in a document called out content exclusion statements. Students will not have to memorize the Krebs cycle. They will not have to memorize all the steps in the Calvin cycle. Now that doesn't mean that they are not to be familiar with those cycles, but if they're given like a diagram and ask them questions, that's a whole different thing than asking kids to memorize things. And you know, what content is just above and beyond the level of the course. So these big ideas 1, 2, 3, and 4. Don't have to read them verbatim, and what I did, if you just want one word, big idea, think evolution. Big idea 2, energy, cell processes. And for example, here's the idea in here, homeostasis, which is an overarching, one of those important core principles and concepts. Big idea 3, information, storage, transmission, essential to life processes. So in here, information transfer could be DNA and RNA. It could also be information transfer on cell signaling processes. What else is in here? The nervous system would be a good example of information transfer. Big idea 4 is all about interactions. And when you look at the terms throughout this document, biological systems, when we looked at these big ideas, we really, to make them really all-encompassing, are there concepts, are there those enduring understandings in these big ideas that can be applied from the molecular level to the cellular level and all the way up through the population, community, and ecosystems levels? So for example, if we're talking about information transfer between cells, if you're talking about information transfer between, you know, individuals in a population or between different organisms in a community. So we looked at all of these, again, construction, you know, that you can find information in there all the way from molecules and cells all the way up to a very large grain size would be the ecosystem level. So I thought it would be helpful if you've not seen the document, and I know you're going to rush home tonight or tomorrow and first time you can get to your computer and a printer to print out that curriculum framework, I'm just going to take a little section of it and show you how we can unpack it. Bio-Rad definitely provides support for these big ideas. And if you haven't checked out the Explorer website and the tools that are available, that's another little sub-homework you can do. So for example, if you look at the P-Glow Bacterial Transformation Kit, you'll see this in a little bit later down the road. Definitely this lab will address those concepts in Big Idea 3 that encompasses genetics and genetic engineering. If you look at their new biofuel enzyme kit, and again, we're thinking all the way from the, you know, cells and molecules and cells all the way up to interactions between organisms, that you can actually apply to concepts in Big Ideas 1 about evolution, Big Idea 2 about cell processes and energy, and then about to Big Idea 4, which is all about interactions. So to unpack one of these big ideas to see how, you know, this thing all fits together, I've just taken one little section as a sample for tonight. And, you know, as you were going through this, if you could think of what you're currently teaching in your class now and how your class can map to just this little snippet I'm going to show you. If you're brand new, this will give you an idea. Okay, now I can see at least I have one little unit started already. So, add a big idea 3. That's living systems store, retrieve, transmit, and respond to information essential to life processes. One of the enduring understandings under there would be 3A. Heritable information provides for continuity of life. I don't think there's one of us that does not think that these, this enduring understanding is probably something you want the kids to leave your class with and have a good understanding of that. Under this, 3A1, here's your essential knowledge. DNA, and in some cases RNA, is the primary source of heritable information. If you were to go to another essential knowledge statement that maybe would be 3B or 3C or 3D, maybe you would be talking about, horizontal gene transfer, or you would be talking about viruses, but this specifically now is going to talk about DNA as a source of heritable information. If you break this down a little bit more, a little tiny snippet would be, where does genetic engineering come in? And we do want students to leave knowing that genetic engineering techniques can manipulate the heritable information of DNA and, in special cases, RNA. And how you're able to plan an activity and a teaching moment around this concept, this little piece of knowledge. Illustrative examples would include electrophoresis, polymerase chain, transformation, restriction enzyme analysis. And because these are illustrative, you don't necessarily have to address all of these and have to do a lab based on all of these. So you can pick and choose. And as we're going to see, one of the learning objectives really thinks that kids should be familiar with at least 2 of these. So if this was a test question that was being written, it would be, from the following examples, please describe 2. So if students are not familiar with polymerase chain, they're not going to be marked off, and they could go off and say, but I do know what electrophoresis is and how it's used, or we did DNA fingerprints, we did profiling. So this is how you're going to unpack to a much finer grain size the information in the big ideas. So if you look through the curriculum framework under those big ideas, and it's sectioned very nicely, you indeed already have the outline of the enduring understandings, the essential knowledge pieces, the illustrative examples, and if there could be some kind of exclusion statement down here, that will also be included for you. So now if your question is, what are students expected to know about genetic engineering, instead of just the vague thing they have to know about genetic engineering, you can sort of see how this is going to be unpacked. These science practices, and where I'm going with this is because in a few minutes we're going to see how to make those learning objectives that are going to fuse a piece of content with one of these science practices. You are probably already having kids do these, but maybe not using this language when, creating objectives for an activity or for a lecture or for a unit in your syllabus. But I don't think there's anything in here, and when the first iteration of these things were written in a language that was, I don't think any teacher alive, unless you actually wrote them, could understand them. So we made sure that the language of these science practices was as teacher-friendly as possible. So, one, the student can use representations and models, can engage in scientific questioning, they can plan and implement data collection, they can then analyze data, they can work with scientific explanations and theories, maybe revise them. This number 2 really is important for some very good reasons. The student can use mathematics appropriately. And one of the things that the commission absolutely argued for, and it was to the College Board, that if you don't grant this request, we're going to be very, very unhappy, that finally AP Biology is coming into the fold along with chemistry, physics, and environmental science, that now the course will be a higher emphasis on quantitative application using mathematics. Students now will be able to use calculators on the exam, so questions that are data-based can be more realistic. So for example, in the past, if they were asked to solve a genetics problem with the dihybrid and they're expecting a 9-to-3 to 3-to-1, the data was so contrived that it would almost fit exactly a 9-to-3 to 3-to-1. Now the data may be a little bit more messy for them to work with, a little more messy, but also a little more realistic. In light of this, also like chemistry and physics, we are now going to have a biology formula list of calculations. That doesn't mean that over the course of the year, students are going to have to be familiar with all of those. But those areas of the curriculum framework, for example, that talk about Hardy-Weinberg, or they talk about being able to do chi-square analysis, the students will be able to work with this formula list, and it's in the curriculum module all year. And then when they come to the exam, they will need a little calculator. The proctor will check these. It's just a little tiny, you can get them for $1.50. The little calculator only has to have the 4 basic functions and a square root, because, you know, we're not going to have students where they can pre-program and graphing. It's not that complicated. But this is a big, big change that I know teachers have been clamoring for a very long time, that the use of mathematics in biology is just as important as the use of mathematics in the other disciplines. So if you want to expand these science practices, and again, at the end of that curriculum framework, that document that you're going to download, print out, and carry around with you to the beach and on a plane and anything else, you know, fishing boat you do this summer, you'll see that under these science practices, they're going to be fleshed out a little bit more, that the student can work with scientific explanations. What does that mean? For example, under 6.4, and I think there's about 5 of these, the student can make claims and predictions. Making predictions is very hard for kids. You know, okay, we see this data. What do you think may happen to this lake 10 years from now? And then give some, you know, justify your response. Also, Science Practice 3.0 about students engaging in scientific questioning. One of the big things is always encourage your students to ask questions. Don't necessarily provide easy answers. We're teaching a generation of kids that they have instant access to information. They no longer have to go to a card catalog in a library. They don't have to, you know, they can just, Google is a great tool. Unfortunately, I use that in my classroom a lot. But when kids often ask me in class, you know, do you know? I just say no. Let's do a little investigation. What tools do you have? And then really through, you know, activities and labs, encourage them to ask questions, and then another question, and another question, and another question, because asking questions and investigating answers really is at the heart of science. So these things called the learning objectives, and do not panic when I tell you that there are, I think, 144 of them. There used to be more. These will be spelled out totally in the appropriate sections in that curriculum framework. And again, these provide the foundation for the laboratory experience and, most importantly, for the exam writers. They are working with the same learning objectives that we see and we are familiar with. How they are constructed, they are going to merge a specific piece of content with one of these science practices. So this science practice will be the task that students are asked on the AP exam. Explain, make a prediction, justify your prediction, describe, revise this model. And I think the important thing to remember, these learning objectives, I guess you can do this, but it's highly discouraged, and that's why I put the little frowny face. These are not to be used as a checklist of content. We've asked, teachers have asked us in the past, does this mean if there are 144 days that my class meets, on day 1, I should cover learning objective 1. On day 2, I cover learning objective 3. No, that's not what they're about. In fact, they are interconnected across all the big ideas. And so if you look at those 4 big ideas, think of that big umbrella that all of the course is interconnected. It's not like it's designed that, you know, quarter 1 you only address evolution because that's big idea 1, and then quarter 2 or the end of your, you know, first semester, then you go on to big idea 2. Everything is interconnected. And we're going to show you then, how you can design instruction around these learning objectives, and especially the test and the lab experience. So remember this little frowny face, not to use these as a checklist of content. Okay, I've done that, learning objectives. My kids know it. They'll never see it again. We can move on. So how you're going to construct these learning objectives. And actually, they've been done for you, but you can certainly get an idea. If you remember this little formula, a learning objective is a piece of content, specific, merged with a science practice. That equals a learning objective. So if we go back to this piece of content that we saw about 6 or 7 slides ago, that I took out of Big Idea 3 of the curriculum framework, genetic engineering techniques can manipulate the heritable information of DNA and in special cases of RNA. That is a piece of content. So the idea of it is, what kind of question, what do you want them to do with that piece of information? So the learning objective 3.5 is going to include this piece of content with Science Practice 6.4, which included justify the claim. So the student can justify the claim that humans can manipulate heritable information by identifying at least 2 commonly used technologies. Another learning objective that fits that same piece of content is the student can pose questions about the ethical, social, or medical issues surrounding human genetic disorders. This piece of content, that there are ethical, social, and medical issues surrounding human genetic disorders, science, biology, stem cell research, cloning, whatever you have, the student, this is required that they really have some idea what kinds of issues are out there, and the student can start posing some questions. As we're going to see, a student could ask, who should have access to your DNA profile? Should it be an insurance company? Should it be the college admissions office? We don't have answers to these, but let the kids start asking some of these questions. That will be their task. So using some of this information, I have just pulled a couple of my activities that I do. And so you can take your current curriculum. You can do the exact same thing. Or if you're starting out, I would hope you have, are working with a mentor or something to get you started here, but you have the curriculum framework. So Ingrid mentioned these course planning and pacing guides. These are on AP Central. There are 4 of them. 4 different authors from very different schools with very different school schedules and different approaches. I happen to be a micro to macro teacher, that I start with the chemistry and work up to ecosystems. But if you work in the opposite direction, one of these 4 course planning and pacing guides has your name written all over it. So the one that I did, a little bit of background about my school. It's a suburban school. It's a private school. We have about 800 students. And so when I do this Unit 4, and we're also on a pretty traditional course schedule, even though great timing that it's going to change this September. But there are 8 periods in the day, and on any given day, the student can meet for either, 6 or 7 periods. And, it's a rotating schedule. That happens to be what my school is, but one of these course planning guides is based on one of the teachers had a more modular schedule, so that was planned out. So it's not only the course planning, but also how long are these activities going to take? And, we're going to address that question tonight, too, because a big fear that I think all of us had, or even have, are we going to be able to get through all this? But then remember that first or, second slide I gave you, how content has been reduced to increase breadth. So in my course planning guide under Unit 4, the little section is called From Gene to Protein, and this is where I do DNA and transcription and translation and the operon and all that good stuff. But my little section on genetic engineering addresses 2 questions. And if you really look at these, it's just putting the learning objectives that I showed you in a more student-friendly language. So by the end of these activities and then the labs that support these, I hope my students really do have some answers to these kind of questions, and I would also hope they would have more questions that we could include down here as being essential. So I just picked a couple activities that students in the past are very fond of, and they've begged me, please, whatever the changes are, do not throw these things out. And you can see how, I sort of, have a, degrees in biology, but I also went to a liberal arts school, so I love music, and I love art, and I love theater, and I love, because I think what AP Biology, as I see, it really attracts so many different types of students. I have students that probably will never set foot in another science classroom. I have students that are definitely going to be doctors and scientists and bioengineers. I have kids that are heavily involved in the theater program. So I really try to open my class, to tap into their talents and my passions and their interests, and there's going to be something for everybody. But when students come back to me after years, away from college, and they look at this activity called A Day in the Life, and you can certainly see where my inspiration came from my son, who has a very rare form of Down syndrome, a 1421 translocation. And, seeing his, following his journey through life, you can see that, I think, students, when they get into human genetic disorders, let's make it real to them. Let's take it out of the textbook and describe a day in the life of a teenager afflicted with a, either a single gene abnormality or a chromosomal disorder. However they want to present their projects, a story, a journal. In my classroom right now, I have oil paintings that were done. I have ceramic statues. One day I had a girl come in with her cello and really came up with just an amazing background song as she had a poem about cystic fibrosis, of all things. It's amazing if you give the kids a little bit of freedom where they can take this, far more than I could possibly do as a teacher. But then a certain thing, remember those essential questions here for this unit have to be incorporated into the project. They have to really start identifying and start asking some questions about some of these ethical issues. So embedded within this project, I am addressing these two learning objectives. 3.26 with that science practice that asks students to explain connections, and then back to the science practice 3.1 that the student really can start asking some questions about ethical, medical, or social issues. And it really is amazing what the kids come up with. And I think sometimes the hardest thing for us to do as teachers is to give up that control. I don't talk about genetic disorders. I don't lecture on this. We do activities, and the kids, when they have the responsibility and they have to teach others, everybody learns, including me. Another activity, if you have not seen the film Gattaca, it came out in 1997, and I'm starting to realize that some of my kids, we're getting to an era where they may not have even been born yet, but they really find this film provocative. First of all, they love films that they know that have been released in the theater for what they call normal people, non-biology, AP biology students. And so this film that came out, Gattaca, in 1997, really foreshadows contemporary issues about genetic manipulation. This was just released right when the Human Genome Project was really cranking up. And they're reflecting on an idea explored in Jurassic Park that just because science can do something doesn't mean that it should. And how students want to address this and share their reflections, I use these mini posters. If you guys know Brad Williamson, he uses these at KU. He's just an advocate of using mini posters where kids make like mini science fair presentations, so it's not just they're turning in a paper for me to look at and give them a grade, where you establish in your classroom or on a table in the hallway where students can take a tour of each other's work, and then using little Post-its, they can critique, they could ask questions on the Post-its to the authors, and it really generates a lot of good engaging discussion and debate by kids. The lab investigations. If you remember 4 words, observe, explore, question, and investigate. Do the same things with your students. Observe them, explore ideas with them, encourage them to ask questions, and help them investigate answers. Observe, explore, question, and investigate. Tonight, you are observing this PowerPoint presentation. We are exploring the curriculum framework. I know you're going to have lots of questions, and then I guess for homework you can investigate some answers. But I think, this is really, really important that when students start acting like scientists, they start thinking like scientists. And now you see the reason why those 7 science practices are so important. Because by having the kids, address those science practices, work with those science practices, they will really get an appreciation how science works. Okay,
we're going to take just a moment here to do a process check and make sure that you all understand that if you have questions for the presenters, either myself or Julianne, to please send your questions through the Q&A function. If you have any technical issues that you're experiencing, please use the chat function to the host for help in those situations. We also want to remind you that this presentation will be available to you all. We will be sending out an email with a link to the recording of the webinar as well as a copy of the slides. Thank you, and back to you, Julianne.
I'm going. Okay. So if you look at the guiding principles for the labs and the lab program and the new lab manual that the College Board has authored, just like the curriculum really had its roots in that 2002 report by the National Research Council, the same thing about the labs anchored in America's lab reports. I've kept this, short and sweet, that students should be involved in inquiry. And in a minute, we're going to look at really what inquiry is. There's many types of inquiry. I did not know that until I started this project. The College Board has promised absolutely that schools and school districts, we know that budgets can be an issue. These labs that are in the new lab manual, and again, they're exemplar labs, they're not required, but none of them require new major purchase of equipment. And for the audit, every school and every course must have a minimum of 8 hands-on lab investigations. That's 2 per big idea, or 25% of your instructional time. And as we're going to see, these labs can be taken directly from the new lab manual, or if you have favorite labs that you've been doing for a very long time, we're going to show you how you can modify those to fit these requirements and the guidance and the flavor behind the new lab program. So the new lab manual is called AP Biology Investigative Lab Manual, an inquiry approach. A lot of people behind this, a lot of authors, reviewers, a lab vision team working with the universities, working with science professional organizations. The lab manual will have 13 labs that are inquiry-based. They will either be exciting brand new labs, or if you really like the 2001 lab manual, even sometimes it's called the Dirty Dozen, you can modify your labs from that lab or from other lab sources or labs that you have created. And within this new lab manual, for each big idea, we have modified a lab from that 2001 lab manual to make them more student-directed, inquiry-based. All the labs, again, will have a greater emphasis on quantitative skills, the application of mathematics. There are both student and teacher versions of the manuals, and you can access them right now on this, again, very important website on AP Central. So what is inquiry? I didn't know, but there are 4 levels of inquiry: confirmation, structured, guided, and open. When I first, years ago heard inquiry, I just envisioned a class where resources were absolutely limitless, that you're just going to turn 40 kids loose in a lab and say, find a question and design an experiment and carry it out. We wish we had the resources for this, but most of us are probably going to be in what are called guided inquiry, that you're going to guide the teachers, maybe teachers will guide students, that you'll present a question, but then let students design and use experiments that they have designed and selected procedures. Try to maybe get a little way, away from the cookbook, do this, do that, where even the answers, there's like a right or wrong, and when you look at their presentations, you see something and, okay, they got it, they got it, they got it. So most of us are going to be in this area right here. Hopefully, as we all become more comfortable with the changes, slowly but surely we may give kids a little more freedom to do a little more, where they come up with the questions and then they can, have a little more free reign kind of thing. So the benefits of inquiry are really, really important, and I think this bullet is probably the most important, is that every time the kids do something, they're going to start asking new questions about that. As they go about with their experiments and with their investigations, something is going to pop up and they're going to ask you, why is this happening, or do you think this could be? And your answer should, I don't know. That's a very good question. What kind of an experiment could you design to maybe investigate an answer to your question? You know, at some point you can't have one lab going on for, 12 years, but certainly, some of these labs in the new lab manual, you can get them started in August or September. 3 minutes a day, 4 minutes a day to do a little bit of, data collection, and you can extend these out depending upon how you, as the teacher, really want to, run your lab program and what resources and time, time is available to you. So if you modify a current or favorite lab, again, we're not throwing out the baby with the bathwater. No one is saying, I'm sorry, you never ever can do any of those labs from the old AP manual again, or you cannot use those labs that you designed and you have really worked on for years. As long as you align those to the concepts, the science practices, and those learning objectives in the curriculum framework, you are good to go. You are good to go. So if you look at the format of the new lab manual, and this I'm really looking at the student version, each new lab will have a more extensive background information section with information, a lot of activities that students can be assigned for homework, or they can collaborate on Facebook or whatever networking site the kids are all seem to be on. They start off with questions to immediately engage students and suck them right into the lab. The learning objectives for students are stated in very friendly student language. A student is not going to know offhand what does it mean to justify a claim. They're going to say, explain your results and give reasons for, your explanation. In the investigation section, there's getting started, there's a procedure, there's analyzing results, there's tips. It says, your teacher may ask you to put your results in the form of a table or a graph, or your teacher's going to give you the freedom to design, how do you think is the most effective way to present your results, what's the most effective way to communicate your results. And then the big section in here, designing and conducting the investigation. So once the kids, you have a pretty good foundation that they know the techniques and the actual skills in the lab that they're going to have to use, then they can go in and conduct their own investigations. And then time permitting, there's a lot of areas of questions either for wet lab investigation or research projects. Where can you go from here? So this is all very new. So the look of the new lab manual inside. Also in that student version, and especially the teacher's version, there are so much good information in the introductory chapters. In fact, one of the chapters was about implementing quantitative skills and practices more in the lab program. We have really simplified that because out of that was born an actual quantitative skills guide that Brad Williamson and John Junk, a really great group of people, actually have written a document that will be available now to give teachers even more suggestions how they can make, applying quantitative skills to the lab program. So to give you some examples, I've taken just 2 labs. That, again, will support those questions that I was, this mini unit that I was taking my kids through genetic engineering. So what we've done in the new manual, we thought it was good to separate the 2 biotechnology labs, because in the 2001 lab manual, they're both in there in Lab 6. But remember that these technologies, students don't have to do all of them. They don't have to do transformation and PCR and gel electrophoresis. It was the idea of at least 2 commonly used technologies. So I will have my students doing the bacterial transformation lab, and then, of course, they will be doing a DNA fingerprint. So these 2 labs, this one will fit this learning objective, this science practice, and also will fit this learning objective with the skill about the students being able to predict how changes in genotype provides a variation. And notice the word here, evolution. Even though these learning objectives come out of Big Idea 3 that includes genetics, this lab also connects to some of those absolute essential concepts in Big Idea 1 that's all about evolution, that the phenotypic variations are subject to natural selection and evolution. So once I have a clear, understanding that my kids are able to do a transformation lab, and here's where in the lab manual, you as a teacher are going to have some freedoms. If your students are already familiar with certain techniques, maybe from a previous course, you may not have to start at the very beginning of the lab. If they've already done a transformation lab, you may be able to ask them, they can skip and get right to work and then get on to the independent investigations. At my school, for 70% of our students as juniors, AP Biology is the first and only course they're going to take in high school in biology. They've had a little bit of life science in the 7th grade, but I certainly, cannot consider a middle school course a prerequisite. So these are questions that students, as they've worked through the transformation lab, these are actual questions that my students have come up with and, said, can we investigate some of these questions? For example, this one. If you, have done the transformation lab, there's that part of it where you have to heat shock the cells, and then you have to, put them for so many seconds on ice. The students have always asked the questions, what about if we heat shock them for more than 45 seconds? I wonder if we don't put them on ice. Will the experiment work? There's their question, and my question is, I don't know. Can you design an experiment to figure that out? They're always curious about the satellite colonies, whether they've been transformed. And, as you sort of progress, they're going to have more questions that you really can open up a little bit more for them. Biorad kits to support Lab 8 will include the PGLOW Bacterial Transformation Lab. And then really, if you really want kids to get into that depth of understanding, if they're familiar with transformation, they're also familiar with gel electrophoresis, have them combine them. Because this kit, the PGLOW SDS Page Extension, really combines these two different labs into one. So that would be a really good one for those kids that have had some familiarity, and how can you take it to that deeper level. And again, these kits can be found on the Biorad Explorer site. Lab 9, here's your biotechnology restriction enzyme analysis of DNA. All of these labs in the new manual start with, we hope, with some kind of provocative question to really get the kids thinking about things. And this one, their answer is immediately, well, of course, I own my DNA. It comes from my cells. And that's when I say, well, can you envision a Gattaca world down there where maybe along with your SAT scores and your college transcripts and your letter of rec that your college also wants your genetic profile? So this one we're going to have kids start with a forensic mystery to get, engage them immediately. If they've already done a DNA fingerprint, they can jump right into this part, and the teacher can take this as deep and as difficult as they want. But again, as the kids are doing this lab, they really are, and they certainly don't know that they're working towards these learning objectives that in their version are spelled out in a more simplified version. Suggestions for independent investigations. This little mystery starts off where the kids arrive to their AP Bio class. The classroom is in disarray and their teacher's missing, and there's some blood drops on the floor. You now get to play along because you get to provide samples of DNA collected at the crime scene. So you can have DNA from the teacher, you can have DNA from a piece of bubble gum that a student has left under a desk. Maybe the teacher next door has come in, or maybe someone has come in with a scraped knee looking for a Band-Aid. And then when the kids work through this, they get to, based on their results, they get to write their conclusion. They get to communicate this. The kids sometimes, for me, do videos with this. They do the PowerPoint presentation. They write out, a script for CSI or whatever they're going to do. And also, of course, there are Biorad kits to support all of these labs. I also include this one because I think what kids, especially if they haven't done this before, they love to extract DNA from their cheek cells. They can even make a little necklace. My kids have said, gosh, a great way to make money for the biology department. Why can't we go to the San Diego State Fair, City Fair, and have a booth where people can extract DNA from their cheek cells. Students actually went with me once to a meeting of an organization called Sisters in Crime, and we had about 80 people in there, and we set up the little lab stations, and my students were like the lab assistants. Everybody in the room got to extract DNA from their cheek cells, and it was a really, really cool afternoon. But again, these BioRad kits definitely support this Lab 9. So at the end of all this, when I've done the interactive assignments, they've done the Day in the Life project, they've done the labs, they've looked at Gattaca, how well are they understanding these two concepts? How are they able to address those two questions that we started off, 2 weeks ago when we started talking about genetic engineering? And this is just one suggestion that I have done in the past. But, sometimes this is actually the summative assessment that I give them. Instead of giving them a traditional multiple choice test, as long as they can give me that information that, shows me that they have a pretty good understanding and a deep understanding of these questions, sometimes I really get creative with what I ask them to do with that information. So speaking of tests, they are going to take what's called the new and improved AP exam in 2013. And it's going to look much different than the one that they took last week. And I have some sample questions. In June, if you go to AP Central, keep checking. The College Board is going to release a full version of a sample AP exam, so that would be something you would love to get your hands on. And so when you start putting your old, your unit test together and writing your test questions, you can have some samples of what the new test is going to look like, smell like, and taste like. It will still have sections 1 and 2. Currently, there are 100 multiple choice questions. That number has been reduced to 63. And you may say, well, wait a minute, how in the world can we cover all this content in only 63 questions? They're pretty packed, and they're going to be a little different looking. And in a second, I'll show you what one's going to look like. This is a brand new, very novel idea. There's going to also be 6 grid-in questions, mathematically based, where students have to come up with a mathematical answer. Maybe they have to figure out the value for Q squared or 2PQ using their calculator, using the formula list, and then they literally grid in their answers on a, looks like a little Scantron, the Scantron sheet. Section 1 will be 90 minutes, 50% of the weight of the exam, and again, here's where students are able to use calculators and the formula sheet. Section 2 is also going to look a little different. I mean, currently there are 4 constructed responses, or what are, we call free response questions, but now we're going to 8, and you should think, oh my goodness, how are kids possibly going to get through this in 80 minutes? But they are now broken up into what we call 6 short responses and then 2 of the more traditional long responses. Because if you even look at the 4 free response questions that have been asked in the past, there's multiple little sections in there that some are worth more points than others, where some require an answer that the kid's going to, if you gave them enough time, they'd be writing for 3 weeks on photosynthesis. And then, one little thing that they just have to, calculate, water potential or something. So these are going to look a little bit different. So, sample multiple choice question. In the curriculum framework, it is required that students have an understanding of the differences between anaerobic and aerobic respiration. So the learning objective, the question that was based on this information, comes out of the learning objective that asks students to be able to make a prediction about the differences between anaerobic and aerobic respiration, and also justifies that prediction. And so here would be your answer choices that would include the prediction and the justification. So this is a little more packed than these questions used to look like in the past. The grid-in, here's a sample. Here would be a little bit of data in an isolated population at Hardy-Weinberg equilibrium. The question would be, how many of the tasters are heterozygous? Again, the student will have a calculator. They'll have the formula sheet. And I think this is really important because with how kids round up and round down, a range of answers will be accepted. So for example, and I'm just, hypothetically, certainly not the answer to this question, but let's say for whatever question is, a rate of a reaction, you were looking at 0.51 and the kids had 0.49 or they had even 0.54, there will be a range of acceptability that kids can grid in and, get credit for the correct answer. So I think that's a step in the right direction. An example of a sample short free response question, it will tell students in a short paragraph of 3 to 4 sentences, describe how the graph, so here's the science skill where students are given a model, a visual representation, and then asking to evaluate that data to answer the question kind of thing. Teacher resources and support. There's so much out there. Go to AP Central, go to Bio-Rad Explorer, homework, print out a copy of that course and exam description, check out online. It's in PDF. You can just look through the lab manual. I think on the College Board store you can buy the student copies. Eventually you'll be able to buy the teacher manual, I think, this summer. We've also included answers, sample answers to questions in the teacher manual for a little bit of guidance. But again, many of the labs, there is no correct answer. The listserv, that we had communicated with each other for a very long time, disappeared at the end of April. It's been replaced by the online teacher community. Here is the website. You'll have to log into that. I think it is the same password and login that you would do for the audit. There's valuable teaching strategies. There's sample exams. There's blogs. There's question and answers. And, part of the commission, we knew that these changes involve every single one of us. There are almost, 9,000 teachers teaching AP Bio. How do you get the word out? How do we learn from each other? And we're literally all in this together. It isn't just the designers of this. It's not the people who make the presentations or write the test. We are part of this online teacher community, and we basically said if the College Board cannot develop this kind of resource, there is no way that these changes in this course could possibly be implemented. Coming soon, probably by the end of the summer, is another great project that the College Board has, the AP Biology Innovation Site. A lot of teaching strategies, sample test questions that students are going to be able to work through to see where they are in their understanding at the beginning, the midpoint, or do they get it. And then again, Bio-Rad, please check out Bio-Rad, and I can't tell you how much I appreciate them for, coming up with this idea to do a webinar from all of us, another way to get the word out. So some frequently asked questions. I think I've addressed all of these. Through the presentation, yes, you can use alternative labs as long as they fit the requirement. No, you do not have to purchase a lot of expensive new equipment. Use what you have. None of these labs require every single student to have their own computer or iPad. You can do them with a projector and one computer, or in a computer lab, or for homework, a lot of the background information can be done. What else did I anticipate? If students come to your class with a solid set of skills, they've had a good year in biology, they've had chemistry, maybe they've had physics or physical science, it's your choice. It's great if you would do all the labs in their entirety, but you may see, my kids have already, done a background on osmosis and diffusion, so they can certainly skip to the more, independently design part of things. Make the class your own. Use your neighborhood resources. When I do the animal behavior lab, instead of using fruit flies or pill bugs, afternoon, I take the kids down to La Jolla Cove where there's a population of harbor seals. I take them down, we're pupping, and we do a thing about animal behavior, maternal instincts, and then the seagulls show up, and there's some, all sorts of critters down there, the tide pools. So we do a very interactive kind of thing. So use that park across the street, or my kids even once designed a lab based on just kindergarten kids on a playground. And it's amazing the behaviors that they came up with. New and improved, it's not going to take longer. It has been piloted many, many times, studied with your typical syllabus, The timing will be about 65 to 70%, and the rest of the time is yours to do whatever you want, to add that extra project, to see that extra film, to go on that field trip, to dive into a lab a little bit deeper. And all of these validation studies and pilots support that. The expectations are reasonable for a typical 2-semester course in introductory biology. Bio-Rad, again, supports all of these big ideas: evolution, energy and cell processes, genetics, genetic engineering, big idea 4 about interactions. So again, all, Bio-Rad, other resources, everyone is behind this. We're all part of this wonderful, amazing kind of journey. And talk about violating my no longer will you hear me for 20 minutes. I almost don't have a voice left, so again, I can't thank you enough. You can get me, email me. I'm at the Bishop School, thedallasj@bishops.com. You can ask questions through Bio-Rad. You may see me if you go to APAC this summer or some of the other conferences. But communicate with us, communicate with the College Board, and again, I hope my point tonight was to relieve some anxieties. And, I'm excited about this, and I hope we can all be excited and engaged. And in many ways, I think we're becoming students again. I love it with some of these new labs that I sort of have looked at, just to join a lab group and literally be one of those kids in that little group and, see science, that excitement that they have because they're seeing things for the very first time. So I thank Ingrid, I thank Bio-Rad, and especially I thank all of you on, a Wednesday evening for joining us here. So Ingrid, I'm going to turn it back over to you.
Thank you, and we appreciate your expertise and insight, Julianne. We have many questions, and we only have about 5 minutes left to be able to address them. So I want to let all the participants know that if we don't get to your question live here, we will be following up in the email when we send out the recording link and the PowerPoint presentation, and we'll have a list of answers to your questions as well. So the first question that I have, Julianne, is a participant saying, I've heard that some colleges are considering not accepting any score level for credit because of what is perceived as reduction in rigor or content of the course. Can you comment on this?
My comment is that the perception is actually quite the opposite. That because now it's transparent, the colleges know exactly what students are learning. And remember that validation study. This was sent out to colleges and universities all across the United States, from northern Maine to southern California, and over 60 chairs of college biology departments and professors. And notice that on all of these, the lab project, the commission, the review committee, the AP biology, development committee, the college community has been just as involved in this process as the high school and the college board. And so the pilot studies, the validation studies, and there were many, many studies that the college community, in fact, many of them said that there is more depth of instruction in the AP course than at many of their Biology 101 classes.
Thank you, Julianne. The next question is, are the chapters that were not listed, we're referring back to slide 9, where you list the chapters in the Campbell AP Biology text. So are the ones that were not listed going to be eliminated from the AP content focus?
No. I was just, that just slide was a sample of how some of the content has been reduced, not all encompassing. But for example, they just said, okay, Julie, for the new course, what you do now, there's 55 chapters in the book. Where do you see the big cuts? So I just gave that as an example. But for example, no longer will students have to specifically study the anatomy and physiology of the jellyfish. But if you want to do that, that's fine. That's an illustrative example that you can, make your class your own.
Great. So the next question that we have is, will the new exam use specific illustrative examples listed as choices in the curriculum framework? If so, won't this advantage those students whose instructors made the right guess?
No, because if they're illustrative, and I can't even tell you you know, when an exam question is put out there, first of all, no question will ever appear on the real AP exam unless it's been piloted by students. It's gone through many iterations. To write exams takes a 3-year process. So ETS has been working with the development committee for 3 years now to write these exam questions. And this is where the expertise comes in. When you see a question and you see that that content is illustrative, the only way that question will make it if there is enough information in, you know, the scenario to the question or the question itself, or there's a diagram that it's fair game for everybody. You know, we know that not every piece of content is going to be addressed, but if it's not required, if it's not marked as required in that curriculum framework, it's not going to be there. For example, a few years ago, one of the 4 essay questions was based on kids' knowledge of muscle contraction. And you think of all of the things that kids could study all year, and wow, I spent, you know, 10 minutes this year on actin and myosin. That's the kind of stuff that will not be on there because that's, first of all, the muscular system is no longer required. It's illustrative. And so especially those free response questions are going to be more generalized, more encompassing, and more areas for students to, you know, focus in on what they've actually learned rather than what they did not learn.
Great, thank you. The next question is, my students frequently do not come to me with enough background to know what questions to ask. How do I teach scientific questioning? How do I meet all these learning objectives when I need to invest so much time reteaching lower-level bio concepts?
One thing that the College Board cannot require, they cannot make, you know, require schools and school districts, but my school is rather unusual because for my students, this is their first-year biology course, but it really is recommended as a prerequisite by the College Board and just about everybody that AP Biology should be a second-year course. So students should be coming into AP Bio with some kind of, you know, knowledge and skills base. I know, for example, like at my school, not everyone who signs up for AP Bio can just walk into the class and take it. They have to be recommended by their chemistry teacher. They have to have, you know, they can't be failing math. They have to have writing skills. So questions, you know, start simple in the classroom. You know, as you're in the front of the class. Every day I start off on the board, question of the day. This is where we're going to get discussion started. This is where we're going to go today. And then I take the last 5 minutes of class, and then I go around the room and, you know, in a couple minutes tell students to, with the people sitting next to them, come up with some other questions that you still have based on what we did today. So, I mean, I appreciate the question, and it's difficult to answer that because I know a lot of kids are coming in with a lot of different backgrounds, and we would love it if everyone had a full year of biology, you know, before tackling the advanced level, but the College Board cannot dictate that.
Well, thank you, Julianne. We're out of time, so we want to thank all of the participants for joining us. I hope we answered a majority of your questions, and again, we'll try and answer the remaining ones in our follow-up email. And thank you, Julianne. Again, we really appreciate this.
And thank you, everybody, for spending time with us tonight.
Thank you. Have a good evening, everyone.
Thank you again for joining us today. When we close this session, a survey will pop up on your screen. We appreciate any feedback you provide. You may now disconnect. Have a great day.